Hi, in this screencast, we're going to learn about a concept called LD50 and a thing called dose response curves. It's something that, I don't know, is something that you probably haven't heard about. It's kind of cool and it's not that hard. Let's get going. Okay, so for any substance we can think of, if you get exposed to enough of it, it can kill, right? So even water, if you drank enough water, your blood would thin out and your organs would fail, and so any substance can have a toxic effect or a toxicity level, and we want to compare. Obviously, water is something that doesn't scare us, and DDT does, and so we want to come up with a relative amount of uh, danger associated with these different substances. And so in 1928, a scientist came up with this concept called the LD50, or how much of a dose does it take to kill 50% of a population. And so we've got to keep in mind is that there's genetic variation within all organisms, right? So within a species, there's just genetic biodiversity. So not every member of a species is equally susceptible to a poison. Some will die with small exposures, some will die with more exposures. And so this is a way of getting a handle on that and quantifying it in a meaningful way. And do you like my dead bugs? It took me a while to draw those. Okay, so again, LD50 is defined to be the amount of substance it takes to kill 50% of a test group. So I expose a test group, half of them live, half of them die. The amount that I gave them is standardized in terms of milligrams per kilograms. And then that's what I report as my LD50. Now, you have to keep in mind when I do this, I'm probably going to be giving it to test subjects that are not exactly 1 kilogram in mass. Some of them might be smaller than that, like in the case of lab rats, or more than that, maybe in the case of rabbits. So I have to standardize it, and so we standardize it to how many milligrams of dose per kilogram of body mass. So let's just say that we did an experiment, and I know this seems awful, but we need to do this in order to make sure that we can ensure the safety of people and just the environment itself. We need to know, we need to be able to quantify toxicity values. All right, so don't judge. Okay, well, I understand if you don't like that they do this, but they do this. Okay, so let's just say that we determine that a dose of 66 milligrams of a particular substance will cause the death of 50% of a population of lab rabbits that were exposed to this, and these rabbits have a mass on average mass of 3 kilograms. So we say then as well, for the LD50, this would not be 66 milligrams because that's what it takes to kill a 3 kilogram rabbit. So instead we'd say, well, it was 66 milligrams per 3 kilograms of rabbit, which gives us a standardized LD50 of 22 milligrams per kilogram. Now, does that mean that 22 milligrams per kilogram would kill another species, say humans? Probably not, but it is a rough estimation. And let's be clear, like we need to know this, but we don't want to be evil. We're not going to test it on humans. We're not going to kill all kinds of species just to get a more precise value of this. It's a general estimate, but we would just assume then that 22 milligrams per kilogram would apply to other species, including humans. That's just an assumption we make, even though we know it's probably not quite correct. Now, it's worth pointing out that LD50 is specific only to one type of an exposure, and that's called acute exposure. That means a one-time massive dose, well, not massive, whatever it is, but a one-time dosage of it. This is not about a chronic accumulation of it over time in your environment. It's like you got exposed to it and you either lived or you died. So we need to be clear about that. So this is not about bioaccumulation. This is about acute exposure. Now, because this is about acute exposure, we had to make sure that our test subjects get subjected to it all at once. And this usually means that we're going to have to administer it orally, by with a like just squirt it into the mouths of these animals, or by injection, or sometimes they can absorb through their skin. Now, it's worth noting that some toxins are absorbed by the act of breathing. So that's true for a lot of pesticides. It's how they kill, and human exposures to some things that are breathed in. So in that case, we talk about the LC50, which is the lethal concentration, and it's basically determined the exact same way, which we'll get to in a minute. But in this case, instead of it being milligrams of chemical per kilograms of organism, instead we want to put it in terms of the concentration in the mixture that's being breathed in. So if it's humans, we're talking about what is the concentration of this toxin in the air. So it would be in parts per million concentration in the air per kilogram of organism being exposed. Or if it was fish, it might be parts per million in the water per kilogram of fish. Okay, but it's the same basic idea. It's just a different exposure methodology. Now, because LD50 varies between species, we'll never really know exactly what it takes to kill a person. You know, again, fortunately, we're not going to do these kind of studies. But what we do to try to make sure we have some sort of standardized methodology that will allow us to refine over time, so we tend to use a standard lab rat or lab mouse. And these are the two main organisms that we use. And they've been they're almost genetically identical to each other. And so as we learn more, we can refine our models because we're always comparing it to a very defined, there's very low genetic biodiversity in these lab animal populations, and that just helps us get it allows us to extrapolate better. Okay, and so. But basically what we'll do is once we establish an LD50, we'll say, look, we get it in milligrams per kilogram. We'll just assume that that's true for humans as well, even if we don't really know. So let's just look at an example. Let's just say that we determined that the LD50 for a particular substance is 22 milligrams per kilogram, and that's what we find in rats. Well, what's a human likely to die? So we'd say, well, the LD50 for a human is going to be the same thing, we will assume. And so then we just say, well, what is the mass of the person? What's the 60 kilogram person who got exposed? So we say, well, 60 kilograms. And then we just say, we'll use dimensional analysis. We'll put the 22 milligrams in the numerator, put the kilograms in the denominator, and we get 1,320 milligrams. Now, what that means then is that a 60 kilogram person exposed would have a 50-50 chance of surviving. Or similarly, if 100 people got exposed to it, then 50 of them would probably die and 50 of them probably would not. That's what we mean by LD50. Now, one of the very useful things about LD50, arguably one of the most useful things about it, is that it allows us to quantify relative toxicity of different substances. So when a manufacturer of some new compound comes along, they say, okay, I have this new product and I want to, I think it's a good herbicide, let's say. Well, okay, we've got to do a lot of things. You got to show that it's not so durable that it'll bioaccumulate. There's a lot of other things you have to do. But one of the things you have to do is you have to come up with an LD50 for it. So you have to say, okay, so how toxic is it relative to other things? So that's really one of the powerful aspects of LD50. So let's just compare relative toxicity. So let's just say, and I made these numbers up, okay? Let's just say that for DDT, we find it has an LD50 of 40 milligrams per kilogram. PCBs, we come up with 20, and nicotine 4. And by the way, nicotine is very deadly. When people smoke cigarettes, they're getting trace amounts of it. But it used to be used as a poison. People put on assassins used to put nicotine, pure nicotine on a knife, stab somebody, or even with a needle and they die. Anyway, it's very toxic is my point. Now, so from this here, though, we could say, remember, 40 means it took 40 milligrams of this to kill 50% of the subjects, but it only took 20 of this. So it's you got to remember, the lower the number, the less it takes to kill, the more toxic it is. So therefore, based on this, I would say, well, from this data, I would assume that PCB is twice as toxic as DDT because it took half as much to kill. Similarly, nicotine is 10 times as toxic as DDT because it only took 1/10 as much. And it's 5 times more toxic than PCB because it took 5 times less. So that's how we make those comparisons. Just keep in mind that the smaller the value, the more toxic it is. Now let's just talk about how we determine it. So you may very well, on their test, have to do a lab with this. And it's an interesting process, and it makes a lot of sense. And it's good because it gives you lots of opportunity to practice what you know about experimental design. So first of all, you got to have a lot of organisms to do this. And I know people get upset, the idea of killing all these rats, but this is basically what is necessary in order to protect people and the environment. So we take a large group of organisms and we divide them into groups of, say, 20 or 100 or whatever. So and now we're going to expose each group of, let's just say, 20 rats to varying levels of toxins. So we're going to take one group and there'll be our control group. We're not going to expose them to anything, just saline solution. We're going to take another one and we're going to give them a low dose. And we take the next group and give them a slightly higher dose, and then a slightly higher dose, and a slightly higher dose, and maybe like significantly higher doses, okay? And so we're just going to have each of these groups. And then what we do is, first of all, we had to administer all at once, because remember, this is to find out about acute exposure. So we inject them or squirt it in their mouths or rub it on their skin, or if it's LC50, put it in their breathing environment. But then what we do is we say, okay, we're going to have a predetermined time. Let's just say 24 hours or 42 hours, whatever we decide. We're going to say after a certain amount of time, we're just going to count how many of these organisms in each group are alive and how many are dead, and come up with a mortality percentage. What percent mortality was there? If they're all dead, 100% mortality. If none of them are dead, 0. If 50% of them dead, 50% mortality. That's how we're going to do this. Now, and then we're going to graph this, and we're going to get what's called a dose response curve. And from this, we can then interpolate this dose response curve to figure out what we're really after, which is, what is the dose that would kill 50% of the group members? Because that will let us calculate LD50. So let's see how we do this. All right, so let's just say we did this study here. Okay, I'm just using small amounts of it because I don't have a lot of room on my slides here. Okay, so here's what we've got. We're going to take a quarter kilogram rabbits, so 0.25 kilogram rats, rather. Okay, and we're going to do this. We're going to divide them into 1, 2, 3, 4, 5. Well, really, there was a 6, 1, 2 that didn't get any, so I didn't put down the control group. Okay, so we're going to have a sample size of 20 rabbits in each test group, and we're going to expose this group to 0.1 milligrams of toxin. We're going to take this group and we're going to expose them to 0.5. We're going to take this one, we're going to give them 1 milligram. We're going to take this group, we're going to give all of them 10 kilograms. We're going to take this one, give them all 100. Wait that prescribed amount of time, and then count the living, count the dead. So we found, presumably for our control group, which I didn't put on here, which got 0 of this, we should find that all 20 of them are alive and we have a 0% mortality rate. Well, look at this. We gave them 0.1 and we still had a 0% mortality rate. When we got to about 0.5 milligrams, 2 of them died. So that gives us, 2 out of 20. So that gives us a 10% mortality. When we gave them 1%, 1 milligram, we got 40% of them died. We gave 10 milligrams, 70%, and so on. Now, just looking at this, I can tell that 50% is somewhere between these two values here, right? Somewhere between 1 milligram and 10 milligram, half of them died. And if you look, 1 milligram was getting pretty close to it. So it's probably not too much more than that. Right? It should be somewhere maybe like around 3, I'm guessing. Okay, but the best way to do it then would be to use what's called a dose response curve. But let's just, for right now, let's just assume it's 3, okay? Well, 3 milligrams is not my LD50. 3 milligrams is what killed 50% of rats of this mass. Remember, I always want to express it in milligrams per kilogram. So I'd have to say 3 milligrams, which seems to be about what it took to kill half of these rats, divided by the mass of the rats, gives me 12 milligrams per kilogram. That would be my LD50. All right. Now let's just see what the dose response curve would look like. A dose response curve looks like this. It looks a little bit like, or quite a bit actually, like the carrying capacity curve. So it's a logistic curve. So the idea is that at first, none of them were dying. Oops. Okay. Someone's trying to tell me something. None of them died. And then here's our data. And this data isn't perfect, okay, but it just gives the idea that we have the scatter. We're just going to try to fit this logistic curve through it like this, because they always have that shape to them. And from this, then we want to try to interpolate, well, what was the amount that caused a 50% mortality? And I apologize, my graphs aren't that great. I'm trying my best, but it's like I'm not a professional. So here you go. So what I find then is that I go to find the LD50, I go to 50% mortality. I go across until I hit my interpolated logistic line. And then I go down. Now, it's worth noting that in most cases, we're going to have a very wide range of doses that we give. We've got to give enough to get more than 50%, or we're not going to know what LD50 is. And so often what that means is we're going to end up having to use, like I have here, a logarithmic scale here, which is a little bit difficult to interpolate, but you guys have done it before, and you can do it again. I think we did this with survival curves. Okay, so I go over to my logistic curve, I drop down, and if this is a logarithmic scale, if this is 1 and this is 10, that's probably right around 3, okay? So I now have 3 milligrams of toxin killed my rats, so that means it would be 12 milligrams because they're 0.25 kilograms each. All right, and so okay, that's what I just said here. Okay, so basically, okay, so if this is 3 milligrams and my test subjects were 0.25 kilograms, I'm going to express it as a standard milligrams per kilogram. I get 12. Now, the next thing I want to talk about is this part here, because this is actually one of the more interesting parts of it, and this is what we call the threshold. So there was some exposure that took place before any response was noticed, which is what's called a dose response curve. And when it first deviates from 0, that's the threshold limit, okay, or the threshold dose. And this threshold dose is very useful for purposes of doing toxicology studies, because we want to know how much can people get exposed to without causing harm. Is what we want to know. And keep in mind that these are mortality; these are dead. We don't want dead. We don't want any dead, right? But we also want to know, well, how much would be harmful? So this is how we make that determination. We don't expose people to it and just see how many of them get hurt. We expose lab rats. We get a curve like this. We find the threshold, and from that threshold, we can determine what we consider to be a safe dose, a safe human dose. So here's how we do this. So I look at this threshold. Remember, this is a logarithmic scale. This looks like it's about 0.3 milligrams. So 0.3, anything below 0.3 milligrams didn't seem to do anything to any of my test subjects. So what I would do then is I'd say, okay, well, but my test subjects are 0.25 kilograms. So I need to divide by this value here, this threshold value of 0.3. I need to divide it by the mass of my subjects. And that gives me a threshold value, a threshold dose value of 1.2 milligrams per kilogram. Anything below that should not cause any mortality. But I'm not worried about mortality. I'm worried about harm. Okay, so what I do is I use this equation here. So what I do to find the basically what we call the safe human dose, I'm going to take the threshold dose, which we just had from here, which was 1.2 milligrams per kilogram. I'm going to take that threshold dose. I'm going to multiply it by the, say, standard mass of a human being. Okay. And that would tell me, okay, well, this is how much would start killing people. But I don't want to kill people. So what I'm going to do is I'm going to divide by a safety margin. And that safety margin can be anything from, say, 10 to 1,000. Hopefully we're going to go close to 1,000 and really err on the side of caution. And so we just put that in here just to make darn sure we're not killing people. And from this, we're able to establish a safe human dose, that people can be exposed to this and we don't expect them to be harmed from acute exposure again. So let's just try this here. So the safe human dose is what we're looking for this particular toxin. We found that the threshold dose was 1.2 milligrams per kilogram of rat mass. We say, well, a human being, let's just say on average is 70 kilograms. We divide that by 100 and we come up with this. A human can be exposed to 0.084 milligrams per kilogram per day, and we would not expect them to have any ill effects as a result of this. Okay, so that is it for LD50 and dose response curves. I told you, it's really not that hard. It's kind of interesting, and I think you'll do well on it when you encounter it on the test. And I've got to use this different method, so it's going to take me a while to turn this off. Sorry about that.